Hello, good afternoon, and thank you for the invitation to participate in this patient day. Um, I want to make sure that this is recording, um, but let me start by sharing my screen with the presentation that I would like to give today. Um, Thanks again. Thanks to the group at Hopkins. Thank you, Hugh and Crystal and everyone at Hopkins for inviting me and giving me the opportunity to participate. Um, I would like to present to you some of the results that we have obtained recently and give you a little bit of an overview of where things stand from the point of view of the basic research. And hopefully you will see also some translational research that we're doing. And there will be at the beginning some very basic concepts that I am sure you know. Then we'll get a little bit more complicated, hopefully not too much, and trying to bring you into the cellular function. And then also, hopefully, you will see some potential for translation in what I will present today. So let me start with something that you know. The heartbeat results from the synchronized contraction of millions of cells. Each one of the single cardiac myocytes of the cardiac cells that are muscle cells is able to contract. And for each one of them to contract, there has got to be a functional coupling inside the cell between the electrical part of the cell that gives a jolt and the mechanical event of the cell in which the electrical energy is transduced into a contraction. Now, arrhythmogenic cardiomyopathy is an interesting case in which the electrical part of the heart and the mechanical part of the heart, both sides can present some very serious conditions. And that has brought us into trying to ask ourselves what is so peculiar about arrhythmogenic cardiomyopathy in the level of the single cell. Indeed, when you have a heart in which there has been some structural damage, there are conditions that are not only cellular that can facilitate the generation of arrhythmias. And of course, the deficiency in the contraction of the heart is an organ effect and not a single cell effect. But what is happening at the single cell level in between the electrical part of the cell and the mechanical part of the cell? And is there a connection between the two? So what we have done for actually a number of years is to take on the idea that in terms of the arrhythmia, in ARVC, or at least in our case, we have focused on plakophilin-2 deficiency. There is something about the mutations or the loss of plakophilin-2 that makes the electrical part of the cell not to function properly at the single cell level. And this started a number of years when we thought that maybe plakophilin-2 is not only making Velcro between cells, but also has some functions inside the cell that are important for the heart to contract properly and for to generate an action potential properly. So we have done a number of studies on the relation between desmosomes and arrhythmias at the cellular level, but actually there have been no equivalent studies at the level of the contractility of the myocyte and the mechanical side. So what we have done is to investigate whether the ability of a single cell to contract is affected by a loss of expression of plakophilin-2. So this is what we use and that we will be describing results from in the next few slides. This that you see here is a single adult cardiac myocyte from a mouse that has been placed on a dish and is going to be paced with wires that are not touching the cell, they are outside. And what you can see in this video is that indeed when we pace the cell shortens. There is a non-linear, but there is a relation between how much a cell shortens and how much force it can develop. And so we have been studying this shortening of the cell as a surrogate of how well the cells can contract at the single cell level. The other thing that we have been doing is trying to understand what exercise has to do with plakophilin-2 deficiency. And so what we have been doing is taking some mice and putting them to run on a treadmill. And this video is not running, but you can see a picture of the treadmill here. And in one of the lanes, there is a mouse. This treadmill moves, and the mice train in this treadmill for 1 hour for 6 weeks. So 6 weeks of 1 hour running. And so we have 3 experimental groups. The mice that are control, and then the mice that are deficient in plakophilin-2. Now, what is this deficiency in plakophilin-2 that we study? What we do is that we have genetically engineered mice so that they are born and develop with their normal plakophilin-2. At whatever time we decide, we will inject them with a compound that will genetically knock out the plakophilin-2 gene. So at the time that we inject them, the mice will start losing the expression of plakophilin-2, and this will be done only in cardiac myocytes. So we're going to knock out plakophilin-2 in young adult mice, and we're going to do it only in the cardiac muscle cells of adult mice. And we follow what happens from that day 0 on. And we do this in mice that are sedentary, but we have also been doing it in the mice that are following the treadmill program. 14 days after the mice are injected, there are no changes in the structure of the heart. What you see on this side is a cross-section of the heart, free wall of right ventricle, septum, free wall of left ventricle in a slice of the heart. And here we have exaggerated the presence of fibroid material, of collagen. And you can see that both free walls and the septum at this point do not present much collagen. But 1 week later, 21 days after we injected the mice in order to knock out the plakophilin-2 gene, here you can see that the free wall of the right ventricle is now showing damage. It is thinner, and on the other hand, the left ventricle is still quite okay, as well as the septum. So at this stage, we have a phenotype of a cardiomyopathy of right ventricular predominance. And if we test these animals for arrhythmias, they are arrhythmogenic. So we have a phenotype of an arrhythmogenic right ventricular cardiomyopathy. Now, I always say this: we do not recapitulate the disease because, frankly, and with all due respect to my friends and colleagues, none of us can recapitulate human ARVC. The reason is, first of all, that any experiment in mouse has an obvious difference from condition in human. But the other is that when you take, for example, cells from a human and you culture them to become cardiac myocytes, those cells are not mature. And it is only cardiac myocytes in a dish. And the disease is far more complex, far more complex than what can be presented in a dish with immature cardiac myocytes. So we don't recapitulate the disease. What we do study is the endophenotype, is the phenotype of a heart deficient in a gene that, when it is mutated in the human, causes a disease. So we are able to establish some hypotheses that are based on observations that occur in either mice or in cells in culture. With that preamble, this is how we have been studying the contractility of cells that don't have plakophilin-2. This again is a single myocyte from one end of the myocyte to the other. And you can see that myocytes have these vertical lines of a darker shade. Each one of those lines limits the single contractile unit of a myocyte, which is called a sarcomere. So many repeats of the same unit, of the same molecular components that are called the sarcomere, multiple units are present along the length of a cell. So what we do with the experiments that I showed you earlier is that we measure with a computer a particular area of the cell in which the computer is going to track the position on every single one of these gray lines. And then is going to average all of them and is going to plot the length between those lines, because when the cell contracts, those lines come together, the length of those between those two lines as a function of time. And each one of these red lines is the electrical jolt that will cause the contraction. So you can see here that when we pace the cell, the lines come together, they come closer, so the length of the sarcomere is less because the lines are coming together to each other. That is the contraction, and this is the relaxation. The cell relaxes, those lines come back to their normal position and are ready for the next contraction. So this would be, so to speak, the diastolic period of the cell. This would be the systolic period of the cell, in broad terms. So what happens when we average the results from these contractions? What we have found is the following. This is an average, right? So this is the length of the sarcomere in percent from control. You can see that the cell shortens, reaches a peak, and then relaxes. When we study the cells from the plakophilin deficient mice, to our surprise, these cells actually contract better. Actually, I should not say better. They contract more. And that is the plot that you see in this red line. And when we tested the cells of the exercised mice, then is the dotted line. They are actually contracting even more. So the shortening in the sarcomere and the shortening of the whole cell is more. And yes, in basic principles, that more shortening would be able to translate in a more intense contraction of the cell. Paradoxically, perhaps, because at the beginning we thought that the individual cell was not going to contract more, it was going to contract less. So at this point, I have to pause. Well, first to show you the compounded results. And so indeed, there is more sarcomere shortening, cells are contracting more. And with exercise, that is even more exaggerated. So how does the cell contract? And what is this issue of the excitation-contraction coupling? Well, we have here a um, model of the elements of excitation contraction. And we're going to follow the fate of calcium molecules because turns out calcium is the great transducer of electromechanical force. Calcium enters the cell through calcium channels on each electrical jolt. In front of the calcium channel, inside the cell, there is this organelle that stores calcium inside. And it is presenting to the outside this channel protein called RYR2, ryanodine receptor 2. Now, the ryanodine receptor is a tricky channel because should be normally closed. When calcium touches its outside, is going to open. Now, I told you that there is a lot of calcium inside. So when that little calcium touches here, the channel will open and it will allow a rush of calcium to get out. And so if we track the amount of calcium inside the cell, you see that the calcium goes up with the electrical event, that is the AP, the action potential. Calcium rushes out, and when it goes out, it goes to where the myofilaments are, where the fibers for the contractile fibers are. Now, the contraction of the cell occurs because calcium gets into the fiber and allows the fibers to slide into each other and bring the edges of the sarcomere closer together. Calcium determines the beginning of the contraction. Calcium determines its end because it is when the fiber releases that calcium that the fiber will relax. Just following the calcium for the moment, this calcium should go back inside its storage space, be ready for the next beat. And to re-enter its storage space, it uses a molecule that requires phospholamban. And some of you may have heard of phospholamban, as mutations in phospholamban can also be causative of ARVC. Calcium can also be extruded from the cell through this molecule. Important thing to remember about it is that when one calcium is pushed out, in order to have the energy to push it out, this molecule uses 3 sodiums in. So the balance is that there is more sodium and therefore more positive charges coming in than leaving. That can depolarize the cell, and if this happens in excess, can create the condition for another extra beat that should not be there. Also note that calcium can be stored inside mitochondria. Mitochondria is the source of energy, so we need good mitochondria to provide the energy for the contraction to occur. And there's got to be the right amount of calcium in the mitochondria. Too much is bad for the cell. So, with all of this intro and this calcium story, what is happening with the calcium inside the cell? In the cells that don't have plakophilin-2, the amount of calcium that is released on every beat is actually more than in sedentary animals. And that is even more so with exercise. With exercise, there is even more of a release of calcium. So what is happening is that there is more calcium being released. Therefore, there is more calcium for the contraction to occur, and that gives us an exaggerated amount of calcium in the cytoplasm. And as I said, more calcium, yes, will give us more shortening, but more is not always better because With too much calcium in the soup, there is going to be too much calcium into the mitochondria, and that is going to intoxicate the mitochondria. And there is going to be too much calcium that this exchanger needs to push out, and that is going to bring sodium that can lead to extra beats. So even if there is more of a shortening of the sarcomere, it is not good for the cell. We asked, though, at least there should be a period in which the heart should be contracting better. Why is it that while this is happening, the hearts don't actually contract better? And the reason is that actually, as this is happening, because the mitochondria are getting intoxicated, the mitochondria are favoring the death of the cell. And so when we measure the amount of muscle that we have in these hearts, the amount of healthy muscle is less in knockout hearts than in control hearts. And that happens both in the RV and that happens in the LV, although to a lesser extent at this particular point in time. So we have this system of excitation contraction coupling. It looks as though calcium is the intermediary that can give us, if there is too much of it, can give us both arrhythmias and a contractility problem. Now, in the center of this release of calcium is, as I said, this molecule, ryanodine receptor 2. And one thing that I did not mention, but is important in the context of what follows, is that the ryanodine receptor in cells that don't have plakophilin-2 happens to be way too eager to spew calcium from its storage space when it shouldn't. So during diastole, these channels have to be closed, and this storage space is not emptying into the cytoplasm. Everything stays inside. But in plakophilin-2 deficient cells, these channels, for reasons that we can discuss later, turn to be too eager to flicker. And as they flicker, they are spewing calcium out, and it just makes things worse. This guy works extra hours, puts more sodium in, depolarizes the cell, more chances for arrhythmias, and the poor mitochondria is getting all of these loads of calcium, keeps accumulating calcium until points in which the mitochondria becomes not able to maintain the normal homeostasis of the cell. So can we stop the ryanodine receptor from flickering? So we took a page from work that our colleague Bjorn Mollman has done studying CPVT, and we tried a drug in the mice, a drug called flecainide. Flecainide is known as a sodium channel blocker, but it is also known as a blocker. He demonstrated to be a blocker of the ryanodine receptor. These are the types of arrhythmias. I just want you to pay attention to these two columns. At day 21 after tamoxifen in our mice, when we give the mice isoproterenol, 60% of them, 6 out of 10, develop during the 20 minutes that followed the bolus of isoproterenol more than 100 PVCs. And the average PVCs, the average of extrasystoles during that period was 600 and some. When we did this in control mice, they did not develop the arrhythmias. So when we gave them flecainide, we did not get any arrhythmia. This is a study that we published already 4 years ago, and came on the heels of this other paper that showed that, just as an anecdotal observation, that use of flecainide in combination antiarrhythmic therapy seemed to have a positive effect in patients with ARVC. And so NIH funded a pilot project for a randomized trial to test flecainide in ARVC patients. And This is a project that is led by Wojciech Zajeva and by Hugh Hopkins, and that has components in other institutions, including NYU. That project, as far as I know, is still ongoing. Now, is there something better? Because flecainide is blocking sodium channels as well, and it can have pro-arrhythmic effects. Well, recently also Bjorn Nolman showed that an unnatural enantiomer of this molecule, verticillide, can inhibit the RYR2 channels, prevent calcium leak, and prevent arrhythmias in, again, a mouse model of CPVT. Now, verticillide is not a drug. It is the natural, not the unnatural, but the natural verticillide is actually used as a pesticide. But the unnatural enantiomer does not have any effects. As far as they can tell, it doesn't have any other effect but to block RYR2. But the toxicology of this compound is still under study. And on the other hand, their lab continues to work on finding new molecules that can block RYR2. In our mice, the enantiomer of verticillide has a very powerful effect to prevent isoproterenol-induced arrhythmias. So in vehicle, 50% of our mice in this parallel side-to-side study developed more than 400 in average extrasystoles during 30 minutes of recording. And when we treated them with verticillide, there was none. Now, I've said flecainide has its problems. Verticillide is not even a drug. So also the Vanderbilt group, under the lead of Bill Stevenson and under a project that also includes our colleague Bjorn Nolman, has been studying the possibility of using dantrolene. Dantrolene is an approved drug for IV injection. It is a very powerful blocker of ryanodine receptors, both the skeletal muscle and the heart muscle ryanodine receptor. And because of its powerful effect on the skeletal muscle ryanodine receptor, is used as an emergency drug in a condition called malignant hyperthermia. What they are doing now is that they have initiated, and it is now listed in clinicaltrials.gov, a clinical trial utilizing dantrolene in patients with ventricular arrhythmias. These are not patients with ARVC. These are, many of them, for what I know, patients with ischemic cardiomyopathy. But what they are testing is the feasibility of using IV dantrolene to study the effects of RYR2 inhibition on cardiac electrophysiology, hemodynamics, and ventricular arrhythmia inducibility in patients with structural heart disease that are referred for VT ablation. So these are patients that obviously are consented for this, in which they are testing before they go in for the VT ablation procedure. They first test the inducibility of the arrhythmias in the presence of dantrolene to see if they have an inhibitory effect on arrhythmia inducibility. So we are now in our lab ready to test dantrolene in our mouse model and see whether we can also have an arrhythmia effect in our mouse model of plakophilin-2 deficiency. So what I leave you with is this observation that we think that this excitation-contraction coupling process that is fundamental for the regulation of calcium, the control of electrical activity through this cold sodium calcium exchanger, and the control of how much calcium comes into the mitochondria. This whole element of the cell is a critical node to understand the arrhythmogenic and the cardiomyopathic events that lead to the disease phenotype. With this, I thank you very much for your attention, and thank you very much for having me. And I'll be happy the day of the session to address any questions.